everyone. My name is Tanisha Vaish, and I'm here to present my independent research project, Plant-Derived Disinfection, a step toward global health equity. But first, a little bit about me. I'm a rising junior at the Harker School from the Bay Area, California. I'm also an aspiring doctor and surgeon and global health advocate. I've scored in the top 30% of the United States of America Biology Olympiad, or USABO, and I've been a national quarterfinalist at the National Science Bee. At Brown University, I've studied cell biology, and at UCI, I've investigated emerging cancer treatment technologies. At my school, I'm the vice president of Neuroscience Club. Additionally, I've earned an honorable mention from the Santa Clara Valley Science and Engineering Fair Association for the research I will be presenting today. So, let's get into it. First, I'll discuss the applications and inspiration for my project. Second, I'll talk about why I chose to investigate the disinfectant properties of persimmons over other plants or fruits. Then, I'll describe my experimental process and the results I found. Finally, I'll talk about future steps and how my research can help millions of people across the globe. So let's start by talking about hygiene gaps, a preventable global crisis. Many classic disinfectants, such as Clorox and bleach, are unaffordable, and 1.4 million deaths, as reported by the WHO, are a result of unsafe drinking water and poor sanitation every year. According to the CDC, another 2.3 billion people lack basic hygiene resources at home. Now, introducing the persimmon-based solution, an affordable and easily accessible disinfectant made using the non-functioning part of a plant and rainwater. This solution could positively impact many people globally. Unlike other common disinfectants used today, it does not have harmful flammable chemicals that can negatively impact its users, making it much safer to use. One of the most common questions I get when I present this project is, why persimmons? Well, persimmons can be found in a variety of environments and climates across the world. Research has also shown that the fruit has exhibited antibacterial properties. Because of this and its widely accessible nature, other researchers chose it as the main ingredient in the natural antibacterial solution. After reading this research, I decided to figure out which specific compound in the persimmon was the most effective antibacterial agent. A natural product, or NP, is defined as anything that is produced by an organism. Persimmons contain a variety of natural products, many of which have antibacterial properties. One of these is tannins, which you may have heard of before, since they are highly present in green tea. They exhibit antibacterial activity without allowing the bacteria to adapt or become resistant. Cytokinins also restrict bacterial growth. Meanwhile, reactive oxygen species, or ROS, are a type of free radical, which forms when a valence electron in a substance is unpaired. These peroxides are very toxic when ingested or present inside our bodies, but they are excellent antibacterial agents. Next are phenolic acids, which are polyphenols that exhibit high antibacterial activity because of a variety of reasons, such as by hindering enzymes and toxins that contribute to bacterial virulence. And lastly, flavonoids. These compounds influence important cellular enzyme functions while having antimicrobial properties. For my project, I tested 4 of these NPs that are all present in high levels in persimmons. First, zeatin, a type of cytokinin. Second, hydrogen peroxide, which is considered an ROS because of its ability to react with different cellular components and cause oxidative stress, damage, or death. Third, gallic acid, a type of phenolic acid. And fourth, quercetin, a type of flavonoid. Fun fact about quercetin, it's also highly present in kiwi fruit, which is another plant that has displayed antibacterial properties. I also tested 2 species of bacteria based on which ones are most commonly found in households, Staphylococcus epidermidis and K12, or non-pathogenic E. coli. I chose household bacteria because of the goal of this project, which is to find a solution that can be used for hygiene purposes in someone's daily life. Some of you might be wondering, well, if the bacteria is non-pathogenic and found around my house, why do I need to kill it? And the answer to that is, although these bacteria are present in our daily environment, and sometimes even on our skin, they can still cause infections when they enter the human body through things like wounds. They can be particularly harmful to individuals with weakened immune systems, such as newborns or those with medical conditions. Each group had a control, which was not treated with anything, and 4 experimental groups for each of the natural products. Next, I measured bacterial growth over time using a spectrophotometer, which is a tool that measures how much light passes through a substance at a given point in time. An important note here is that I did not, however, test a fifth type of NP, tannins. This was because when I added the tannic acid treatment to the bacteria, a chemical reaction occurred with the nutrient broth that caused the solution to become white. Since color affects how much light is passing through a substance, the spectrophotometer gave an inaccurate reading of absorbance for the solution. I'll talk more about this in the future research section. I also tested 3 different concentrations of each NP to measure dose dependence, which means seeing whether adding more treatment would kill more bacteria. I did this to gather even more evidence for the idea that my treatment was killing bacteria instead of other external factors. Within each treatment group, if the only variable is the amount of NP being added, but more or less bacteria are dying, that means that the NP is responsible for the bacterial death. For visual representation, this is what one trial setup would look like for each bacteria. I'd have 5 tubes with different conditions, as you can see here, for each bacteria. I'd have 3 trials like this per bacteria. Here you can see some more pictures of my experimental setup and the devices I used. So, what were my results? The pink line shows how the bacteria were growing before the treatment, and the green line after. In most cases, the green line would be less steep than the pink one, but in some cases, like this one, the slope actually became negative. This shows that the treatment was so successful that it was actually able to kill the bacteria, because dead bacterial cells have a lower absorption than living ones, and the absorption was decreasing. Here's another treatment, hydrogen peroxide, that showed similar results. Overall, the data showed that the treatments were largely successful. Future research would include testing on more deadly bacteria or testing other compounds known to have antibacterial properties. As I mentioned earlier, tannic acid, or the group of tannins, was not tested in my experiment because it did not work with the way that I had originally planned to gather my data. However, this data could still be gathered, but through other experimental methods. For example, a petri dish could be swabbed with the treatment, and the experimenter could physically observe how much bacterial growth was occurring. These persimmon NPs also have anti-cancer potential. I'd love to explore that as well. Finally, the key takeaway. My research contributes to the development of a low-cost, sustainable disinfectant that could improve sanitation in under-resourced communities worldwide. By using natural, locally available materials, the persimmon solution offers a safer, more accessible alternative to harmful chemical disinfectants, addressing a critical barrier in global health equity. Here are my references. I'd like to thank my mentors and my parents, and thank you as well to the Global Health Leaders Conference and Johns Hopkins, and to you for listening to this presentation. Thank you. Here are some ways to contact me, and once again, thank you for listening.